Good day, and thank you for joining me for this Wings for Life lecture today. I have the honor of introducing Dr. Martin Schwab, who I've known for many years and also admired for many years. He is one of those rare breed of scientists that have taken a concept from preclinical development through to early clinical research and mid-phase clinical trials in humans. Dr. Schwab has done this through working and studying at prestigious institutions like Harvard and the Max Planck Institute, and most recently at the University of Zurich. Through his work, Dr. Schwab has gathered many accolades for his investigations, which includes the research that we are to hear about today. He will be describing the long and arduous journey from bench to clinical trials, of which the latest is an ongoing European multinational clinical trial. During this journey, Dr. Schwab has experienced the trials and tribulations of many parts of translating science. That is animal modeling, development and optimization of human antibodies, collaborations between academics and industry, and even of creating a spin-off biotech company. So Dr. Schwab has much to share today, and my best move would be to step aside so that we can hear about his impressive voyage with No-Go A. Thanks, and over to you, Dr. Schwab.
Many thanks, Jane, for this nice introduction. I'm very happy and very honored to give this Wings for Life lecture today here at our Congress. Wings for Life, of course, is a key player as a supporter of research in spinal cord injury, including research in my own lab. You see the declaration of my interests below on the slide. From bench to clinical trials to bedside for a novel therapeutic approach to enhance recovery and also structural regeneration after spinal cord injury is the topic of my talk today. And I'd like to introduce you briefly at the beginning into the biology behind this new concept of enhancing nerve fiber regeneration in the spinal cord and also in the brain of injured organisms. I'll then go through a long series of animal experiments, which I'll illustrate very briefly to you, which show functional recovery as well as structural changes after spinal cord injury when we treat these animals with antibodies which enhance regeneration, and I'll end up with ongoing clinical trials with this therapy. It's known, of course, for more than 100 years that peripheral nerve axons regenerate well after an injury if the scar doesn't inhibit regeneration, and this exact process does not happen in the spinal cord or in the central nervous system in general. Through a series of ex vivo and in vitro experiments, we discovered a long time ago that this absence of regenerative growth in the central nervous system is in fact due to specific proteins which actively inhibit regenerative growth. You see such an experiment here. So optic nerves or sciatic nerves exposed to neonatal neurons and stimulated with neurotrophic factors produce axons. The axons grow through the sciatic nerve explants in these culture systems, and they do not grow into the optic nerve, which is, of course, part of the central nervous system. So an interesting question was why? And the first step was to take these nerves apart, extract myelin, And we found that, to our amazement, peripheral nerve myelin supports neurite growth. This is an overnight experiment here with neonatal dorsal root ganglion cells, and the central nervous system myelin inhibits growth. So the cells that you see here as bright dots survive in these cultures, but they're unable to produce neurites. A long series of biochemical and cell biological studies in my lab and several others have shown that a certain number, up to a dozen or so, constituents are present in oligodendrocytes and in central nervous system myelin, which inhibit nerve fiber growth if you test them in vitro. One of them, NOGO-A, a novel protein at that time, which is a membrane protein in CNS myelin and is absent in peripheral nerve myelin. NOGO-A is the most potent of these factors as far as we know today. In parentheses, oligodendrocytes are not the only structure in the central nervous system which inhibits fiber regeneration. Astrocytes also do it, especially in scar areas, and here a number of different growth inhibitory factors are known. This is the only molecular biochemistry slide that I'll show you in my talk. You see NOGO-A as a membrane protein up here. It has two active sites which bind to two components of a multi-subunit receptor complex. If the receptor is activated by NOGO-A, signaling cascades occur inside the cells. The signaling cascade affects the cytoskeleton, so the movement machinery of the cells and the growing fibers stop moving within minutes. You can observe this in the culture system directly, but there's also a signalosome or a signaling endosome generated, which reaches the cell body of the cell which has contacted this NOGO-A protein and stops the growth machinery on the level of the cell body. Key question, of course, early on in these studies was if we can neutralize or inactivate this NOGO-A protein. What do you see? What happens first in these in vitro systems, and as you will see in the next slide already, also in vivo. So here you're looking at a culture of dissociated neurons. They sit on a culture dish which has been coated with myelin extract from monkey brain. You can do this with different species, rats, mice, and so on. And these cells come from newborn rats. They are unable to produce neurites. They aggregate and survive, but cannot grow. If you treat this culture dish with unrelated, unspecific antibody, the situation is very similar. Few fibers, they are short in this overnight experiment. However, if you treat this culture dish with a monoclonal, monospecific antibody against this NOGO-A protein, many of these cells produce fibers. They are nice and long, sometimes branched. And as I said, this is an overnight experiment. You see this within a couple of hours. So remember that a crude myelin extract from monkey brain contains hundreds, in fact, thousands of proteins, different proteins, and you neutralize one of them, NOGO-A, and you turn the inability of these nerve cells to produce fibers into an ability to produce long neurons. So this is what I meant when I said before, there are active growth inhibitors, and one of them, NOGO-A, is a particularly brutal one. So with neutralizing antibodies, we had a tool to go in vivo and to ask the question, which is of course a key question, is this protein, are these neuron growth inhibitors crucial factors for the absence of regeneration of fiber tracts after spinal cord injury? And here you see an early experiment. The rat has a pump which under the skin, which contains antibodies against this NOGO-A protein. Thin catheter leads into the subdural space such that the antibody is infused into the cerebrospinal fluid. It reaches the entire spinal cord and also the brain. We pump these antibodies for 2 weeks. The pump is implanted after the spinal cord injury. The injury is a Asia grade C type lesion, so a very large lesion, but incomplete. You see the anatomy of this lesion reconstructed from a series of histological sections. Here, for a control animal, these are the microcysts which form. There is a lot of scar tissue, also inflammation, and there's pyramidal tract, corticospinal tract, which sprouts a little bit, but is unable to grow into the caudal part of the spinal cord. This is a sister animal which is treated with an antibody against NOGO-A, and you see that the situation changes significantly in that many fibers now elongate over the remaining ventral half of the spinal cord tissue bridge and start to elongate into the caudal spinal cord, and they grow in a rather disorganized way down the spinal cord. They have terminal branches and arborizations, which you see here, which form synapses with spinal interneurons and motor neurons below the lesion. So these were very interesting findings, strong evidence because it's anatomy. I'll show you a bit more of this. This is again now the micrograph corticospinal tract, pyramidal tract, which is cut here by a scissor cut. These are 2 control animals, mice in this case, and these are animals which have no NOGO-A. So here it's not an antibody, but a genetic deletion that we did, and you see how a lot of fibers come out of this tract and grow into the spinal cord and the tissue bridge, which links to the lower part of the spinal cord. This is how it looks like after an antibody infusion. You see the cut, you see the outgrowth, you see growth cones at the end of these fibers. This is caudal to the lesion. The lesion will be here. You see how the fibers have crossed the lesion and bundle up as they grow down the spinal cord. And these are 2 fibers with their typical growth cones, these organelles through which the fiber finds its way through the spinal cord, and they grow towards the lower end of the spinal cord. This is our experiments with 2 different antibodies, so different from the antibody that you saw before. So it's a rat again and a pump infusion, and you see in these reconstructions how these fibers come over the lesion site and track down the spinal cord. You can measure the distances up to ½ centimeter. We have measured up to 18 millimeters, so almost 2 centimeters distance, which in a rat is of course a very long distance. They do not grow through the lesion because of the scar and the cysts. They grow around it and then down the spinal cord. So after having seen many animals and many experiments with antibody treatments, different antibodies, all of them directed against NOGO-A, and also genetic knockout mice, as I showed you briefly before, we of course asked the question whether this axonal regeneration also reflects on the functional level, whether we see functional improvement in these animals. And we do a lot of different tests with these animals, like open field locomotion, grid crossing, beam crossing, swimming, and even bladder function. I show you 3 or 4 examples of this. This is all published in a series of papers. The first one is an example which is inspired by the clinic. It's rat training on a treadmill after partial but large spinal cord injury. You can track the movements of the joint, reconstruct the movement. You see the swing and the stance phase very nicely here in this animal before any injury. This is after the injury, and the treatment with unrelated control antibody. You see a very disrupted movement. Sometimes they try to make a bigger step, sometimes it's a small step, and very often they just drag their legs. So a very disrupted, non-functional movement cycle. And the third image here is a sister animal of this one here, same type of large incomplete spinal cord injury, Asia type C if you want, and treatment with an antibody against NOGO-A for 2 weeks. You see a very nice stance step cycle here, very little foot dragging, so a very functional restoration of locomotion in this test. Here you have narrow beam crossing, which requires, due to the balance that the animal has to have, a lot of descending control. They are very good before the injury. They can't do it after the injury. And the open columns show the control antibody infused animals. They remain very bad. The hind limbs are dragging or slipping off the beam. The black and the gray columns are 2 different anti-NOGO antibodies, and you see a recovery, which is nice, but is not complete. So this very difficult task recovers partially as a consequence of the regenerating fibers in the spinal cord. This example here is a swim test, and we discovered in these rats, as is well known from patients, that if the water in aqua training is a bit coldish, they get these cramps. And you have a spasm score here, and you see how the spasms develop over time. It takes some time, like in humans, for this spasticity phenomenon to appear. The question was, do we see more spasms because we make the system more plastic and more fibers which grow, or do we see less spasms? The gray columns are the control antibody treated animals. The open columns are anti-NOGO antibody treated animals. You see less spasms. Many animals have no spasms at all, and the spasm score is lower. So what this means is that wrong connections in the spinal cord, which leads to these spastic muscle cramps, are avoided. The system somehow knows how to integrate these new fibers into functional circuitry and a meaningful movement sequence. The most recent studies in our lab address bladder function. So we test urodynamics in these animals. They are awake. These are tamed rats. They sit in this box here. We have an implanted bladder catheter. We fill the bladder, they pee. We can measure this. We have implanted electrodes in the sphincter muscle. And what I show you as the only result from a large sequence of results which were published recently is the sphincter EMG. So we fill the bladder. The bladder pressure increases, the animal is spinal cord injured, the voiding is in small droplets, difficult and slow, typical for highly pathologic bladder. And the main reason why this is so is because you have a simultaneous contraction of the detrusor muscle of the bladder, but also aberrantly of the sphincter muscle, this famous DSD, detrusor sphincter dysynergia. And you see this high sphincter EMG, which should not be there. Here you have the sphincter EMG activity. It's low or at a certain level before filling. During voiding, the sphincter activity is very high, a completely aberrant pattern, and this is after voiding. This is an antibody, control antibody infused spinal cord injured animal, and here you have an antinogo antibody treated animal, and you see the highest activity of the sphincter before voiding. This is the guarding reflex. The sphincter has to avoid voiding. Then voiding starts, the sphincter activity goes down as it should be, and after voiding it is low before the bladder fills again. So also the lumbosacral bladder circuitry is under a certain control of the brain again when we enhance regeneration of injured fibers and also plastic rewiring of spared fibers in these animals with large spinal cord injuries. By the way, the injuries in these animals here disrupt 90% of the white matter of the lower thoracic spinal cord. What is the next step? Obviously, towards humans. Monkeys are closest to us. These are macaque monkeys, cynomolgus. And the model we used in a series of collaborative studies with Eric Rouillet's group in Fribourg in Switzerland is a hemisection model. You see the hemisections here. They are in the cervical spinal cord, and the animal loses hand function completely, especially the fine finger movements, but leg function and bladder control recovers, like in human hemi lesion patients. This is the anatomy 6 months after the injury and 5 months after the antinogo antibody infusion. You see the pyramidal tract, which ends at the lesion because it's transected. You see a sprouting zone, and you see very, very few fibers caudal to the lesion in this control antibody infused animal. This is an antinogo antibody treated animal, treated for 1 month. Acutely after the injury, you see many fibers which enter the caudal cervical spinal cord. So we are at C5 here. This will be C5, C6, and further down. You see how these fibers look like. They branch, they bundle. A very nice anatomical result. Of course, in these animals, you can test hand function very beautifully. For instance, the precision grip here between thumb and index finger, and to our amazement, we found that the antinogo antibody-treated animals, these are these red animals here, recover precision movements of their fingers almost to 100%. Whereas the controls, these are the blue and the green ones, recover finger movements only if the lesions are small. So a 40% transection of the spinal cord gives you a recovery of 60 to 80%, but an 80% or 70% transection leaves you with very impaired hand and finger movements. These monkeys very often have this closed hand here. They are not able to extend the fingers, in contrast to an anti-novo antibody-treated monkey, which you see here. So a series of experiments showed us that, again, different novo antibodies, including a clinical human antibody, leads to functional recovery of the hand and the arm movements in these macaque monkeys. This is the summary of more than 20 years of work in my laboratory at the University and ETH Zurich. You see, you have seen the existence of nerve growth inhibitory proteins in adult mammalian brain and spinal cord. One of them is NOVO-A, and it's a particularly potent inhibitor. Nerve fiber growth inhibitory protein NOVO-A can be neutralized, for instance, by specific antibodies. They are applied intrathecally because antibodies do not penetrate the blood-brain barrier, as you know, and this leads to enhanced fiber regeneration over long distances in the spinal cord. It also leads to enhanced compensatory sprouting by intact spared fibers, and these anatomical phenomena are associated with improved functional outcome in locomotion, in swimming, in bladder control, and also in hand and finger control. Importantly, I have not shown this, but we have a large number of observations on this. We do not see side effects. I showed you only spasticity, which goes down. We see no change in the pain threshold. We see no epilepsies or other things which you could expect when you make fibers grow in the central nervous system. Two additional points. In addition to neutralizing antibodies, you can genetically delete NOVO-A. I showed you one slide on this. You can knock down NOVO-A, again, with genetic techniques. You can block the receptors such that NOVO-A does not activate the receptor anymore in a variety of ways. I've also shown you that there is a post-receptor intracellular signaling pathway. Also, this can be blocked by particular drugs and agents, and in all these cases, you find these two main results, outcomes, namely enhanced regeneration, enhanced sprouting, and functional improvements in spinal cord injured rats, mice, and monkeys. These results over the last 15 to 20 years have been found in several laboratories, independent labs working on this NOVO-A protein, and they have been published in a large number of papers. So this basic science, of course, is a good starting point to now think along the line of clinical translation of these findings. So we are at the moment here in basic research. What you have to do is, of course, produce a drug, an antibody in this case, which can be applied in humans. You have to show efficacy and the absence of toxicological side effects and so on of this drug, and then you move into clinical trials. We are at the moment here in phase 2 clinical trials, and in the last series of slides that I'd like to show you, I'll briefly go over this recent work. So this is these are the steps for the development of anti-novo therapy in humans. We started this with Novartis a long time ago, and a new spin-off company of the University of Zurich, Novago Therapeutics, has taken this up more recently. The target here is a fully human, 100% human clinical antibody which neutralizes human NOGO-A. You have to produce such an antibody, of course, under good manufacturer conditions, which is an enormous effort and also requires an enormous amount of money. We went through toxicological studies. The regulators require 2 species, so rodents and primates, with intrathecal application. We have even done spinal cord injured macaques for one of these antibodies to show that there's no side effects of this treatment with these antibodies. You have to formulate the antibody and then fill it and so on. And you have to get, of course, approval, which in Europe is the national agency, Swissmedic for Switzerland, a European agency, EMA, and for the US, the Food and Drug Administration, FDA. This is one side of what you need. The other side is the clinical network where you can conduct such a trial. A long time ago, with my clinical colleagues here in Zurich, we founded a European network of leading spinal cord injury centers. It's the EMSCI network. Many of you probably know this. There is a North American analog, NACTIN, to this, where we standardize many readouts, diagnostic tools, refined the diagnostic tools. We train the neurologists, the rehab doctors, the physiotherapists, study nurses, and so on. And then we were ready to move into what is first a phase 1 and then a phase 2 of this trial. Here is the phase 1 trial, which was conducted in 2006 to 2012. You find the information on clinicaltrials.gov, as all the other clinical trials also. The trial was conducted in a European multicentric network in a number of European countries. The antibody is called ATI-355 or NG101. We had a large number of acute paraplegic and tetraplegic patients, over 50, all of them ASIA A grade when they entered the trial. The antibody application is initially by pumps, lumbar pumps. Then we found that injections are easier because external pumps are difficult for these patients, which are difficult to treat medically for a number of reasons, which you know better than me. We start early, so within days after the injury, as soon as the diagnosis, severity and level of the injury is clear, which is usually between 5 and 10 days after the injury, we apply the antibody for 1 month with repeated injections or pumps, as I said. Now, the readouts in a phase 1 trial are largely pharmacokinetic and safety tolerability readouts. They were very good. We had no adverse side effects linked to the drug, which is not trivial. This is an antibody into the central nervous system, antibody directed against a central nervous system protein, NOGO-A. No neurological, no psychiatric, no systemic side effects. The half-life in the CSF is short, as is well known for antibodies. It's in the usual range in peripheral blood. And then, of course, the initial cohorts of patients had low exposure for a short time. Then we increased the dose, we increased the exposure to 1 month. And we found that in the later cohorts of the trial, 7 out of 19 patients, which were all Asia A at the beginning, showed conversion to Asia B or C, 1 patient even to D. And very interestingly, these conversions came up late, at 3 months, 6 months, even later. So a time point where conversions are rare, very rare, but also a time point where we know from the animal experiments that the functional improvements appear. They don't appear immediately, they appear with a slow time course. So these results were, of course, encouraging to enter what's called a phase 2 clinical trial, which is the proof of concept trial. So here you are blind, double blind, it's placebo controlled, randomized as is required to show efficacy of any treatment. This trial now is run as a university-sponsored trial by the University of Zurich and the University of Heidelberg. It's an investigator-initiated trial. The antibody is the same, has been donated to us by Novartis and had to be reanalyzed and processed locally here. We have again EMSCI clinics. The money for the trial comes from the EU, so we are bound to Europe, 5 European countries, 14 leading spinal cord injury centers in these 5 countries. We started in 2019 with the trial, and up to now, 73 patients have been treated. We need a total of 114 patients for the statistics, and we will reach this goal in summer next year. Again, you can find information on clinicaltrials.gov, and there's also a web page. The trial has the abbreviation NISCI for NOGO inhibitory antibody in spinal cord injury. How's the trial designed? We go for cervical injuries only. We include this time not only Asia A, but Asia B and C patients also. We stratify the patients according to the severity of the injury within the first few days after the injury. We use the unbiased recursive partitioning procedure for stratifying these patients. The first injection starts in this time window between 4 and 28 days after the injury. We inject over 30 days, every 5 days, so 6 injections intrathecal in the lumbar liquor space. The randomized procedure gives us a 3-to-1 scheme for Verum antibody versus placebo, and we have 160 matched historical controls in addition to the placebo for the final evaluation. The readouts, the primary readout, you have only one, which you have to define strictly at the beginning of the trial. Primary readout is upper extremity motor score. Depending on the severity of the lesion, you have a spontaneous degree of recovery. We want 40% above spontaneous recovery, so 40% better than the controls. Secondary readouts are skin score, locomotion scores, bladder function with bladder diary and the other bladder measurements, and also, of course, pain, and to some extent, also somatosensory potentials. So, as I said, this trial is ongoing, and interestingly, a second independent trial, which is done with antibody-like molecule. It's called NogoTrap or AXR 205 reagent that has been produced in the laboratory of Stephen Strittmatter at Yale, and is a trap for Nogo, binds to Nogo and inactivates it. So here, phase 1/2 trial is ongoing in a number of clinics in the US. It's run by Strittmatter's company, Regenerative Bio. It looks at chronic spinal cord injuries, so not acute like us. It uses a very similar protocol and readouts, upper extremity motor score, hand function, scheme score, and this trial is also ongoing at the moment. Again, it's in its phase 2 part, double blind, so there are no results before the trial will be finished. So interesting times. Keep your fingers crossed for us. Of course, it would be fantastic if we see something, even if the effects are small, then we would have a way to go further and develop further this regenerative therapy, which is an entirely new way, of course, to look at spinal cord injuries from a therapeutic point of view. I'm greatly thankful to my teams. This is the clinical NISCI teams with the leaders of these 14 clinics and their study nurses and study doctors. This is the Zurich team, which has reanalyzed and supplies the anti-Nogo antibody to the NISCI trial. And this is my current research group at the University of Zurich, and the founders which support the basic science work and also the clinical trials. Thank you very much for your attention. I'm, of course, more than happy to answer any questions you would have.